The final learning objective for the gustation and olfaction module, and in fact all of the physiology modules for the semester, is to briefly describe the structure and function of the olfactory receptors. Olfactory receptors are the specialised receptors responsible for detecting odour molecules in the air, or more commonly referred to as our sense of smell. So chemicals that have an odour and bind to and stimulate olfactory receptors are called odorants. Olfactory receptor cells respond to the binding of an odorant by producing a receptor potential. Unlike gustation or taste, where we can only detect 5 tastes, it's estimated that humans can recognise about 10,000 different odours. To make this possible, the nose contains between 10 and 100 million receptors for the sense of smell or olfaction. And these are all contained within a region called the olfactory epithelium. The olfactory epithelium is found in the superior portion of your nasal cavity, along with the superior nasal conchae, which are the little ridges within the walls of your nose, which help circulate your air as you breathe through your nose. So the olfactory epithelium consists of 3 kinds of cells: olfactory receptor cells, supporting cells, and basal cells. The axons of the olfactory receptor cells form the olfactory nerve. The body of the olfactory receptor cell sits in the olfactory epithelium, and cilia, which extend out from the dendrites of the olfactory receptor cell, project into the mucous membrane that lines our nose. The cilia, or the little fingers on the olfactory receptor cells, are the site of olfactory transduction, so taking that chemical signal and turning it into that receptor potential. The supporting cells provide structural support and help maintain the integrity of the olfactory epithelium. They provide physical support, nourishment, and electrical insulation for the receptor cells. The basal cells are stem cells which are located between the bases of the supporting cells. They continually undergo cell division to produce new olfactory receptor cells, which only live for about a month or so before being replaced. Within the connective tissue that supports the olfactory epithelium are also olfactory glands, also called Bowman's glands. And these produce a mucus that is carried to the surface of the epithelium by ducts. The mucus moistens the surface of the olfactory epithelium, but it also dissolves the odorants so that they can interact with olfactory receptor cells and smell can occur. So smell is much like taste, where the odorant actually has to be dissolved in the mucus for it to react or interact with those olfactory receptor cells. So a lot of that might not make a whole lot of sense without an image. So we have a skull here. This is our nose, and the olfactory epithelium sits in the superior portion of the nasal cavity. In our zoomed in image down here, this is the olfactory epithelium. So made up of our olfactory receptor cells, which are this light blue colour. These have cilia which project out into our mucous membrane. We have supporting cells, so these cells along here, which line, nourish, and protect the olfactory receptor cells. And then we have these darker blue basal cells, which produce the new olfactory receptor cells. We also have our olfactory gland, also called our Bowman's gland, and these help secrete mucus. So the mucus is produced in the gland and then secreted down through this duct to help line that nasal cavity. So for olfactory transduction to occur, or for an odour to be smelt, the odorant must first be dissolved in mucus for it to then bind to the olfactory cilia on an olfactory receptor. Binding of an odorant to a receptor on the olfactory cilia stimulates a G protein, which in turn activates an enzyme called adenylate cyclase to produce a substance called cyclic AMP. The cyclic AMP then opens ligand-gated sodium channels on the olfactory receptor cell that allows sodium to enter the cell and causes depolarization. The ability to differentiate between smells is thought to depend on the patterns of activity that arise from the activation of many different combinations of our olfactory receptor cells. Now, just going over that once more on the steps that I'd like you to understand. The odorant must be dissolved in mucus to actually bind to the cilia on our olfactory receptor cell. When that odorant binds to the cilia, it activates a G protein. Activation of the G protein activates an enzyme which produces cyclic AMP. Cyclic AMP opens up sodium channels on the cilia. Sodium can enter our olfactory receptor cell and depolarization can occur. And then finally, after a nerve impulse is generated in that olfactory receptor cell, it travels along the axon of that receptor cell, which joins with the axons of many other receptor cells to form the left and the right olfactory nerves, or our cranial nerve 1. So these axons here will form part of our first cranial nerve. Now, the olfactory nerves terminate in what we call our olfactory bulb. These sit just inside the skull, just inferior to our frontal lobes of the brain. Axons of our frontal bulb will extend posteriorly and form our olfactory tract. Some of the axons of the olfactory tract will extend to the primary olfactory area, which sits in our temporal lobe. This is where the conscious awareness of smell begins. Other axons will project to the limbic system and the hypothalamus, and we're not always aware, at least consciously, of these sensations.